Okay, so welcome back to this next video in which we are discussing ion channels and cancer. Okay, right, so in the previous video what we did is we looked at some principles linking general ion channels to cancer. Okay, and the two main things that we saw here were that cancer cells generally have a permanently depolarized electrical potential difference across their cell membrane. Okay, and this is caused by dysregulated expression of ion channels, and it seems to be very essential in their overproliferation. Okay, indeed, if you stop the permanent depolarization artificially, then you can arrest cell cycling within these cancer cells. We also saw about how electrical potential difference across the cell membrane fluctuates at different points going around the cell cycle. Okay, and this seems to play a role in getting you over certain checkpoints in the cell cycle. And again, ion channels are all concerned in that. Okay, so there's another place where ion channels are potentially linked to cancer. Okay, however, that's all quite woolly. Now what we're going to do is we're going to look at something slightly less woolly. It's still going to be very woolly, but we're going to look at something slightly less woolly, which is some specific examples of where ion channels have been found to be drivers, potentially, in cancer. Okay, so the first one, which is really hopeful as a future target for cancer, is what's known as ether agogo 1. So I'm putting this one first because it is potentially the most important example of an ion channel involved in cancer that we know of at present. Okay, so this stands for ether, and that's the E, A, and then the G is for go-go, and God only knows how it ended up being called that. So it's ether agogo 1. Okay, now its more sensible name is that it is KV 10.1. So it's a voltage-gated potassium channel, and it's in that family number 10, which remember was an example of a family of delayed rectifier potassium channels involved in the repolarization phase of the action potential in neurons. Okay, now the ether agogo 1 voltage-gated potassium channel is a homotetramer of these ether agogo 1 subunits, or these KV 10.1 subunits. So let me draw this out. So just to remind you, you'll have 4 separate subunits coming together to make the full voltage-gated potassium channel, and that will just be referred to as an ether agogo 1 voltage-gated potassium channel, sitting in the plasma membrane like so. Okay, so here is our ether agogo 1 channel. Okay, right. So basically, in the healthy adult human, the ether agogo 1 channel is only found in the central nervous system. Okay, so it's found in neurons in the central nervous system, and outside of the central nervous system, its expression is negligible. Okay, so it's pretty much only expressed in the central nervous system. Okay, so CNS is usually the only place where you find this channel. Okay, however, in 75% of cancers, you find massive great expressions of this channel. Okay, so EAG1, ether agogo 1, is much higher in these cancer cells. Okay, so overexpression in cancer. And this is what's referred to as ectopic expression. Okay, so we call it ectopic EAG1 expression. Okay, because EAG1 should really just be located in the central nervous system, but you're finding it ectopically expressed on cancer cells from all sorts of different peripheral tissues, and this isn't just cancers, within the brain. This is cancers, whatever, from whichever tissues you like, basically. Okay, so that's interesting. Why is it overexpressed in all these cancers, and is it a driver in the cancerous phenotype? Well, what you can do is to investigate that question, what you can do is you can overexpress it in heterologous systems. So let me just explain what a heterologous system is, because it's a fancy term from experimental science which is really important to understand. So a heterologous system means that you can put it into a cell that would normally not have a gene for Heterologous 1. Okay, so you find a cell, you go to a cell culture laboratory, and basically you find some cell line that doesn't have an Heterologous 1 gene within it, and what you do is you overexpress Heterologous 1 in that cell. Okay, so you put in a gene for Heterologous 1 artificially into the cell, and you get the cell to overexpress it. Okay, so if you get Heterologous 1 overexpression in these heterologous systems like this, and what happens is the cells do start to show cancerous phenotypes. And let me just explain this, because this is actually quite an important point. Okay, what happens is the cells show transformation. Okay, and transformation is a term that people often use in cancer biology, and they use it sort of synonymously with neoplastic transformation. Okay, well, with the actual neoplastic process. Okay, but it's important to understand that there is a difference between transformation and actual cancer. Transformation refers to a process that occurs in petri dishes, which we believe is analogous to what actually happens in cancer. Okay, but there is a difference between what's in a petri dish and what's in a person, obviously. Okay, so let me explain what transformation actually means. So basically, if you grow your cells, let's say in a petri dish, so let me just draw a little petri dish here. If you grow cells, usually what happens is the cells behave very nicely. So you use immortalized cell lines, which means that they will divide and divide and divide forever, basically. They have no Hayflick limit. Okay, but they're not cancerous cell lines. There's a difference between immortalized cell lines, which can divide and divide and divide forever, and cancerous cell lines, or transformed cell lines. Okay, so the immortalized cell lines, they will grow and grow and grow, and they'll form a beautiful monolayer on the bottom of the petri dish. Okay, the cells will have nice spaces in between them. Every cell will be happy, basically. It's not overcrowded. Okay, there is normal homeostasis here. They grow to a point and then they stop. That's the important characteristic here. They will grow until they have filled the space that's available for them. But once they have filled the space, they will stop increasing in population now. Okay, so cell division will reduce, and also, if you do still have some cell division, then any overcrowding will result in apoptosis. Okay, so that's comparable to normal tissue homeostasis. The slight difference, obviously, is that these are immortalized cells. They don't have a Hayflick limit, okay, because we've done something to them to stop them having a Hayflick limit. Okay, now, what then is a transformed cell? Okay, well, a transformed cell, if you grow a transformed cell on a petri dish, what ends up happening is they get completely overcrowded, basically. The cells grow and grow and grow, and even though there isn't enough space anymore, they continue growing, and they start stacking on top of one another. So you end up with multiple layers of cells all on top of one another, and they just don't stop growing. They are immune to the overcrowding signals which tell them to stop dividing and to apoptose, basically. Okay, and that, we believe, is analogous to a cancerous phenotype. Okay, so basically, what this really means is that if you put Heterologous 1 into an immortalized cell line, it causes transformation. Whether that's the same as it causing cancer is a question, okay, because in reality, we are not putting it into a normal cell in the first place. We are putting it into a cell that has no Hayflick limit, which is basically part-way towards cancerous anyway, because removal of the Hayflick limit and immortality of division is one of the properties of cancer. Even though I didn't state that in my initial bit, it's one of the ones that, if we were looking at this in more detail, I would have put in. Okay, that was only supposed to be a brief sort of overview of the process. So we're not putting them into totally normal cells here, we are putting them into immortalized cells. Nevertheless, it is striking that it causes this transformation, basically, this malignant transformation. So the immortalized cells have gone now and are now called transformed cells. Okay, so we would call this a petri dish of transformed cells here. Right, okay, so Heterologous, what does it do then? Well, we don't know is the answer. We don't know how it causes these cells to over-divide like this, okay? But we have found the beginnings of a pathway by which it causes something that can't explain what we're seeing here, but can explain potentially why it's helpful for real cancer cells in a human body. Okay, right. So let's discuss this now. Firstly, the first thing to say is that Heterologous is believed to act by what's known as a non-canonical mechanism for it. Okay, now what does this mean? This means that its mechanism it doesn't involve it actually opening and conducting potassium ions through it at all. Okay, when we talk about non-canonical functions for ion channels, we mean that their function doesn't relate to the fact that they're an ion channel, basically. It just relates to the fact that they're a protein and that they can have signaling roles as proteins, basically, that they can act effectively just like any other signaling protein and interact with other proteins. Okay, so non-canonical roles for ion channels means that the ion channel can just act as a signaling protein independent of its conduction function. Okay, so the reason Heterologous has this powerful effect is believed to be that it works for a non-canonical mechanism. It's not believed to involve the fact that it can actually allow potassium ions to move through it. It's believed to be that it's going to interact with other signaling proteins and cause some sort of signaling pathway that then leads to the cells undergoing this malignant transformation here. Okay, right. So, we can't explain why it causes these cells in the Petri dish to do this at the moment, but what we can explain, or at least what's beginning to emerge, is a potential pathway by which Heterologous overexpression in real cancer cells helps those cancer cells, and it has to do with potentially promoting angiogenesis. Okay, so as I say, this can't explain what it's doing in the Petri dish because, of course, angiogenesis has no relevance at all in the Petri dish. Okay, but in real cancer, angiogenesis is very important. Okay, so right at the beginning, we said that angiogenesis is something that will occur to a tumour even without any mutations promoting it. Okay, so it's normal physiology. When cells become hypoxic, they activate angiogenesis signaling pathways, and angiogenesis begins so that you get blood vessels growing into a tumour. Okay, however, if you do get a mutation that promotes angiogenesis signaling, that's actually going to be beneficial, and it is going to give you a selective advantage because more angiogenesis will mean more nutrient delivery to the cancer cells, and therefore the cancer cells will do better than their neighbours who aren't getting as much angiogenesis. Okay, so this is what we think Heterologous is doing. It's promoting angiogenesis. So we think that what Heterologous, so if I draw a little flow diagram here, okay, we think that through non-canonical signaling pathways, the Heterologous tetramer here, or potentially the Heterologous monomer, we don't know that it has to act through the tetramer, okay, but the Heterologous protein, we think somehow through pathways that haven't been completely elucidated, activates a protein known as HIF-1 alpha, and it's the same protein that gets activated by hypoxia itself. Okay, so HIF-1 alpha stands for hypoxia inducible factor 1 alpha. So HIF is for hypoxia inducible factor, and then 1 alpha just means 1 alpha. Okay, so hypoxia inducible factor 1 alpha, and I'll colour in HIF-1 alpha here in turquoise. Okay, and then what's believed to happen is when HIF-1 alpha gets activated by the Heterologous protein, then it's going to translocate into the nucleus. Okay, so it'll now go into the nucleus, and it's going to affect transcription. Okay, so HIF-1 alpha is a powerful transcription factor which changes gene expression basically by altering transcription. Okay, so it's a transcription factor, and the thing that it's now going to majorly change the expression of is a growth factor called vascular endothelial growth factor. So now what's going to happen is the cell is now going to increase its expression of a protein known as vascular endothelial growth factor, VEGF. So this stands for vascular, that's the V, endothelial, that's the E, and then growth factor, that's the GF. Okay, now what does vascular endothelial growth factor do? Well, it's the cry for help, basically. When tissues become hypoxic, what happens is the hypoxia actually induces the activation of the hypoxia-inducible factor, hence why hypoxia-inducible factor is actually called hypoxia-inducible factor. Okay, and this again acts as a transcription factor and increases the expression of vascular endothelial growth factor. And vascular endothelial growth factor now goes to blood vessels in the local vicinity of the cell and causes the growth of blood vessels towards the area that is producing vascular endothelial growth factor. So if I just get another piece of paper and show you this. Okay, just trying to find another piece of paper. Right, here we go. Right, so basically, if we have our hypoxic tissue here, okay, so let's say these are our cells that aren't getting enough oxygen, okay, they're now going to start producing vascular endothelial growth factor, and this will go to little blood vessels, generally capillaries, in the neighbourhood. So here's a little blood vessel here, and what it will cause is it will cause the capillary to start growing sprouts out towards the source of the vascular endothelial growth factor. Okay, so these are now going to grow towards here, and then the idea is that these two separate sprouts here could eventually fuse together to make a functional vessel that can move blood through this new tissue here. Okay, or through this tissue here that is hypoxic. It's not necessarily a new tissue. Okay, so tumours will do this merely when they become hypoxic. Okay, however, if you overexpress the Heterologous-1, that's going to hugely increase the amount of vascular endothelial growth factor that you produce, the amount of VEGF you produce, and therefore you're going to get more angiogenesis than you would just get from the hypoxia, and therefore that's going to help you survive better. Okay, so that would give those cancer cells that have done this a selective advantage over their neighbours, and then they will progress further than their neighbours, and it would progress you on towards the cancerous phenotype. So that's a potential role of Heterologous-1 in actual tumour cells, but as I say, that doesn't explain why expressing Heterologous-1 at too high levels in the heterologous system caused the malignant transformation. We don't know why that happened, okay? We think that it is again for a non-canonical signalling pathway for the Heterologous-1 protein, rather than through its actual ion channel function. Okay, right, so it's just the protein acting as a protein, as a signalling protein, rather than as an actual ion channel. Okay, right. So, why is Heterologous-1 such a promising target then? Well, hopefully that should be clear. It's only expressed in the central nervous system in an adult human, and then it's expressed on cancer cells. Okay, this is brilliant. This is absolutely brilliant because of the monoclonal antibody revolution that we are now having in pharmacology. Okay, so let me just explain what a monoclonal antibody is. So a monoclonal antibody is a mixture containing loads of antibody molecules. Okay, so strictly speaking, it refers to a mixture containing loads of antibody molecules rather than an individual antibody molecule. And it's a mixture containing loads of antibody molecules that are all identical, basically. Okay, so if we pull one of these antibody molecules out now and draw it bigger, it consists of 4 separate proteins. Okay, 2 heavy chains and 2 light chains. So let me colour in these portions in separate colours. So here in orange, here is 1 heavy chain, and here's another heavy chain. And then here in red, we have light chains here. Okay, so this is our antibody molecule. Okay, now at the head of each of the antibody molecules, you have 2 sites which are capable of binding to proteins. Okay, so these are the antigen binding sites of the antibody molecules. Okay, and antibody molecules will be specific for binding to a specific antigen molecule. Okay, so these antigen binding sites can vary between different antibodies. Now, when we produce a monoclonal antibody, this is going to be a solution containing loads of antibody molecules which all bind to a certain antigen, and their antigen binding sites are going to be identical. Now, let me just contrast it to what a polyclonal antibody is. Okay, a polyclonal antibody is a solution containing loads of antibody molecules which are all against the same protein but which are not all identical. Now, how does that work? Well, if we have an antigen here, let's say this is our antigen, there are loads of different parts of the antigen that antibodies can potentially bind to, and all the different parts, and I'll draw a few of them here. Okay, so little facets of the antigen. Okay, these little facets of the antigen which antibody molecules can potentially bind to are known as epitopes. Okay, so when you make an antibody that's going to bind to an antigen, there are potentially loads of different epitopes on that antigen where the antibody can bind. In a polyclonal antibody, you have a mixture of loads of different antibody molecules which all bind to this antigen, but they bind to different epitopes, and therefore their antigen binding sites are not identical. Okay, in a monoclonal antibody, you have loads of antibody molecules which all bind to the same epitope and all have the same antigen binding site structure in order to bind to that epitope. So that's the difference between a monoclonal antibody and a polyclonal antibody. So a monoclonal antibody is a mixture containing loads of antibody molecules which all have identical antigen binding sites and bind to a single epitope of an antigen. Okay, now, monoclonal antibodies are coming in pharmacology. They are taking over because of their incredible specificity. You can use these things to bind to specific proteins and they don't bind to other proteins. Okay? Now, loads of drugs now are monoclonal antibodies and they bind to their target very specifically. Now, the way of recognizing drugs which are monoclonal antibodies is that they have the suffix mab for monoclonal and then AB for antibody. Okay, right. Now, why is the Eferagogo-1 so hopeful? Well, it's only expressed peripherally in cancer cells. Okay, that is absolutely a gift because monoclonal antibodies cannot cross the blood-brain barrier. Okay, so they can't get into the central nervous system. Okay, so I'll put that here. Can't cross the blood-brain barrier. So they can't get into the central nervous system, and therefore, if we were to put one of these monoclonal antibodies against Eferagogo-1 protein into the blood, then it would bind to Eferagogo-1 proteins, and where are those expressed? Only peripherally in the cancer cells. Okay, and it would not get into the central nervous system, and therefore it wouldn't cause any problems with the Eferagogo-1 proteins in the central nervous system. Okay, and that's why this is so hopeful, because it gives us a way of having a molecule that can bind to these cancer cells and only these cancer cells, so a selective targeted therapy basically. Now, this could potentially have diagnostic roles as well as just therapeutic roles. The idea here are that we could attach onto the monoclonal antibody against Eferagogo-1 some sort of imaging molecule, so maybe some sort of radioactive molecule that we can use to diagnose cancer. Okay, and then the idea would be that the monoclonal antibody would go and bind to cancer cells which have the Eferagogo-1 protein on. So if I just draw this, so here is our monoclonal antibody binding to the Eferagogo-1 protein. So this is the Eferagogo-1 protein here, so I'll just colour this in. Here is Eferagogo-1 in purple here, and then here is the antibody molecule binding to Eferagogo-1. Okay, and then if we've attached on some radioactive or other kind of imaging molecule, then what this would allow us to do is image where the cancer is inside you, and that could have highly useful roles in diagnosis. Okay, so this is some imaging molecule, most likely radioactive of some sort. So imaging molecule. Okay, so that could be very, very useful in diagnosis. In addition, we could also attach onto the antibody molecule cytotoxic drugs, and then we could use the antibody molecule to deliver the cytotoxic drug. So the idea here is that you conjugate on some incredible classical chemotherapeutic agent, so some classical drug, conjugate drug, and then this drug is a cytotoxic agent, and then the idea is that the drug will then cleave off the antibody molecule once it arrives at the site of the cancerous cell, because the antibody molecule has taken it there, and then it will be selective for that cancer cell. Okay, it will now be delivered basically to the cancerous cells and kill the cancer cells, but then it won't be delivered to the normal tissues, and therefore hopefully we can hugely reduce the side effects of delivering such horrible classical chemotherapeutic agents. Okay, so that's the potential role of Eferagogo-1 for diagnosis and therapy of cancer. Okay, right, so we'll call it there for this video, and in the next video what we'll do is we'll look at Eferagogo-2 and its role in cancer.